So, good morning, Gaeta, or good afternoon, I'm sorry, good afternoon. Thank you very much to the organizers and all of you for giving me roughly about 10 minutes to briefly walk you through some of Praxis Precision Medicines. So we're a small biotech that are with our headquarters, partially based here, but we also have a growing group in Boston as well. But before I get into the biotech side of things, I wanted to try and frame a little bit of exactly what are we looking at, we as a pharmaceutical company, but also as scientists. As you'll see, we're trying to think of a really big problem. And as Professor Lowenstein said, you know, the brain is huge. It's 3 pounds, so it's not that big when, say, we want to pick something up. But when you start looking at the numbers that we're dealing with, so a trillion is 10 to the 12th, so we have about 10 to the 11th neurons. Now, what I'm not talking about are the glial cells, which you have about 10 glial cells to every neuron. So that puts us well into the trillions inside our head. Synapses are the spaces where neurons come together and speak to each other. And we have 10 to the 14th synapses in our head. And how these neurons speak to each other, they have literally hundreds of neurotransmitters that the neurons pass to each other to get signals propagated. Now, here comes just the simple math, which is if you have 10 to the 14th synapses forming in roughly the first 18 months of life, you know, that's going to be about 50 million seconds, which is 18 months. And now that means we should have about 2 million synapses forming every second during those years of development. It's a huge number, and we can't do it alone. We have to be as part of a team, as part of a community, to try and tackle this. So, of course, leveraging the brains of Steve Petro, and also one of our founders, and also David Goldstein at Columbia, another founder, we've been able to come together and form Praxis Precision Medicines under the guidance of our CEO, Kiran Reddy, physician, as well as a series of highly trained scientists and operations specialists, as well as regulatory engagement specialists. We've been able to form a considerable amount of money that was put together for very specific projects. And what I'm showing you is a few of our clinical programs in depression, as well as epilepsy and pain. This is our website, and you can find us on the web at www.praxismedicines.com. And what I'm highlighting here are 2 clinical programs that we currently have running here in Australia for depression as well as absence seizures. So now a little bit of about one project that we have. I want to step into what's called persistent current. So in certain sodium channels, when you see on the black line, when a sodium channel fires, it opens and allows a stream of current of ions going into the neuron. And on the black line, you see the normal, or the wild type channel, which fires down, does the action potential, comes back up to reach its baseline. And the persistent current, when you have a certain mutation, is the red line. And it fires very similar to the wild type, which is called the peak current. But then, because of the mutation, it has what's called a persistent current. It has a little trickle into the neuron, which is a really, really, really small signal. However, the implication is that it creates a better environment for the neuron to be able to fire again. So we've heard that there are several existing sodium channel blockers that have very limited benefit. So what we can talk about are selective, the hypothesis, the selective block of pathological persistent sodium current without impairing the peak current. So that's that drop in peak which we need to get the signal going, but it should provide enhanced efficacy and tolerability if we're able to get rid of that long red line and bring it back to the black. So SCN2A on the right, there's an animal model that was developed in Northwestern University in Chicago. And this animal model has a very nice signal for persistent current. And on the left is SCN8A, NaV1.6. And this one has a pathological mutation that we've seen in patients. And what we see is, in the black line, that's where you have that's the control, so no drug. And in both models, when you dose, you actually have, that's the red line, and you actually are able to extend survival of the model. So this is actually very interesting because now Praxis 330 is known to block spontaneous seizures in both models. This is data that we can take and start to action on in our projects. So we are a patient-centric organization. What we do is we look at the patient, we do put the patient in our sights to really understand not only what is the patient going through, but what can we do to improve the life of the patient. Our process is, this is, in simple terms, exploratory. So we take a lot of high-risk unknowns and try to answer them in a systematic way so that we get to our discovery stage where we have a much more focused effort in trying to understand and provide a good lead candidate that we can bring into the clinic. And so from there, excuse me, from there, once you get a drug into the clinic, that's where you can start to answer the real unknowns. How do these drugs affect the patient? From there, and I'm putting the cycle on purpose because we can learn from what happens in the clinic, then bring it back to either exploratory discovery, make improvements back into the clinic. But we don't work in a vacuum. And I put a circle there as a porous circle because we need to look at the widening phenotypic spectrum. As more information, more technology becomes available, and we start to understand the genetics of the epilepsy, we start to have a widening phenotypic spectrum. The dynamics in genetic testing, we just heard about that. And right, sometimes as the technology develops, we have these samples that we've been studied in the past. We have a growing number of new genes. Let's go back and look at these, not only the reinterrogations of the genes, but also look at the dynamics such as the trio. Let's get the parents also sequenced to understand where did the mutations come from. Sharing of data is very important, and in the pre-competitive space, we definitely need to share data. We do share data, but we also share capabilities. And then ultimately, the patient's life. This is really important. This is something that Praxis does actively. We need to connect with patients to really understand what you don't see in the clinic. What's in your notebooks? What do you go through? Because there's certain things, small items, that we just don't understand because you see them and they're not seen by anybody else. Very important. So, one more thing. So, someone asked that, how can you learn more about epilepsy and more about taking care of your loved ones? And so there were several points that you could go to learn more about clinical aspects and treatments, etc. So for those of you, which actually it's pretty much everybody, who's super motivated and you want to know more about the basic science, I recommend that you can go to the Khan Academy. So it's a nonprofit, and it's actually a way, me as a scientist, I actually learned economics and accounting. Because it's very important. It's very important in business. But it's a way that little snippets, 15 minutes, to actually learn some about the basic science. So when you hear about protein translation, the ribosome, transcription regulation, actually, you can learn about this at your leisure. And with that, I'm done.